The Clinical and Translational Research Center is a resource used by UNC investigators to aid in the conduct of clinical trials. This facility is located in UNC hospitals. It provides inpatient and outpatient rooms and is staffed with nurses, nursing assistants, and phlebotomists that carry out infusions, timed blood draws, and other protocol-specific procedures. The Clinical and Translational Research Center works in partnership with the Nutrition Research and Metabolism Corps to conduct nutrition research protocols. The team provides meals for long study visits at the research center. They specialize in planning and preparing controlled nutrient meals for nutrition research protocols.
Many investigators come to me with nutrition research protocol ideas for controlled nutrient diets that have never been done before, have never been done on this campus before. So what our Nutrition Research and Metabolism Core team does for the investigator is plan out the diet methods to carry out that protocol and answer their research question.
An example of the type of feeding trial that we specialize in conducting is the choline depletion study. Each meal was planned by a dietitian using ProNutri software and prepared by diet technicians who weighed each food to 1/10 of a gram to achieve precise nutrient control. To control this vitamin, special products and recipes were developed, such as low choline wheat starch bread. This somewhat unusual diet was developed to contain less than 50 milligrams of choline and was served to inpatients for up to 72 days of inpatient stay in order to study the effects of human choline deficiency. We asked each subject to consume all the food served at each meal. If all foods were not consumed by the research subject, the leftovers were weighed back and actual nutrient intake was calculated and recorded.
Our course specializes in preparing weighed research meals for short feeding studies or even single test meals. In some cases, we may control for macronutrients, single nutrients, or multiple micronutrients.
Examples of this include a high-fat test meal to study irritable bowel syndrome or high glycemic and low glycemic load test meals to examine diet effects on hunger and satiety. We prepare sugar solutions used to conduct a sweet taste preference test. We are experienced in preparing formula diets and homogenized food preparations. An example of this is homogenized Braco sprouts. This formulation required procurement of a large amount of Braco sprouts from the same lot number, processing all of the packages of sprouts with water according to the dilution recipe, mixing all batches together, and then weighing each dose of liquid sprouts into opaque containers to be immediately frozen. This process is very important for consistent dosing of compounds found in whole foods. Lately, research focus has been shifting to the conduct of outpatient feeding trials. Recipe development is very important when planning meals for this study design. Not only does the food have to be nutrient controlled, it also has to be prepared in advance, be palatable, easily transportable, and convenient for the participant to consume at home with all assurances of food safety. Study participants come to the research center for study procedures and to pick up their research meals. It is common practice to ask subjects to pick up meals 3 days a week and ask them to return any uneaten food and all food containers for weighback.
Our Corps is continuously developing new diet methods for different nutrients of interest and study designs. An innovative approach to controlling omega-3 and omega-6 fatty acids for a 3-month outpatient feeding trial was developed by our Corps. Instead of providing 100% of foods, the study participants received 2/3 of their daily food supply in unprepared and prepared form. The participants visited the research center every 2 weeks to pick up a 2-week food supply. Food provision was accompanied by diet counseling, educational handouts, and a website offering more than 75 recipes that meet the diet guidelines. This trial required selection of special foods high and low in the specified fatty acids. Food samples were sent to an NIH laboratory for fatty acid analysis, and the research foods, meal plans, and recipes were planned based on these test results. In addition to planning and overseeing the implementation of feeding study diets, the dietitian plays an active role in screening patients for participation in nutrition studies, educating potential participants about the diet study, and helping to identify obstacles that may prohibit the subject from completing the study or adhering to a research diet. Other services that the dietitian provides are diet counseling for those investigators looking to provide standard of care or alter diet through counseling alone. Examples of this are nutrition counseling for patients with eating disorders, diabetes, cardiovascular disease risk factors, and hypertension. The final product that the investigator receives from the team is the diet data. The nutrient data of interest is tracked, cleaned, and electronically sent to the investigator to use in publication. The team also assists in writing the diet methods for grants and publications.
The Nutrition Research and Metabolism Corps provide a unique service required to conduct nutrition protocols and, more specifically, controlled feeding trials. Testing nutrition hypothesis can be incredibly difficult considering the complexities of the human diet and metabolism. Controlling nutrients in diet requires extensive resources and experience to produce precise and accurate diet data. This is precisely what the Nutrition Research and Metabolism Corps brings to UNC investigators.